Hello everyone, Pranam, Namaskar. Welcome back to my channel Indu BIOTECH. Hope you all are well. In the last lecture, you have learned how to run the NCBI BLAST tool and how to enter the faster sequence or gene ID in the enter box. And if you did not see the last lecture, you can see the link has been given in the description box. And if you are the new for this channel, then you subscribe the channel for getting the notification for latest lecture or video. Today in this video we will learn how to interpret the result of BLAST. So let's start. As you know, BLAST is an abbreviation of Basic Local Alignment Search Tool, and it is state to matching a set of program that used to make the alignment between a nucleotide or protein sequence that called query, and the nucleotides or protein sequences within the database referred to as the subject sequences. For the result output, BLAST uses statistical theory to produce a beta score and expect value, that also called the e-value, for each alignment pair query to hit. The beta score gives an indication of how good the alignment happens. So the higher the score, the better the alignment. Right? So you can see on the screen this is the short summary of your query given for this BLAST search. And first, in the job title have given the accession number, species name, and the name of the gene which you have entered for the BLAST, right? This is the program that has given which program has choose for the BLAST, that is the BLAST, and that means you have selected the nucleotide BLAST for the search, and this is the database that you have selected as the nucleotide, and this is the query ID. Query ID means it is the specific and unique ID that is given for every sequences for the every query from the algorithms, right? And this is the description that includes the species names, gene name with the function, and this is the molecular types. What is the molecular types used for the BLAST, and this is the query length. Query length means what is the length of your query sequence. This is the 1423, and this other report, that means you can also see in other format of the results, right? Here you can see the different types of the results is available, right? So first one is the description, second one is the graphic summary of the results, and third one is the alignment, and fourth is the taxonomy. And today we will go for the graphic summary results, and see, if you will see carefully, you will find that the results are in the different color, right? Different color. Above is some color codes. Here is the some color codes. What is mean of this color or alignment score code? If your query sequence shows smaller than 40, that means the similarity 40, that means the graphic output or results will appear in the black color, and your results showing the similarity smaller than 40. If the similarity score is 40 to 50, the graphic line display as the blue color, and your query will show 40 to 50% of the similarity with the database, other database or other species. And if your result showing, that means the graphic line shows green color, that means the similarity is 50 to 80, or the line shows purple color, your similarity or query coverage is 80 to 200. And this is the if your query shows the red color, that means the query coverage or similarity is greater than 200, right? So here is the your query length is 1425 nucleotide base and query coverage is 100%. So you can see, when you put your cursor on any of these line, you can find the information about that species or similarity sequences. So if I put in the first line, this is the our query sequence query, that is the Saccharomyces cerevisiae, gene is ROX3 gene, and this one is the another strain of the Saccharomyces that is also showing the 100% similarity with our queries. And you also can see in the some sequences or some line, you can see the line has the different length. Some one is greater and some is smaller. What is mean of this irregular type of the length? This irregular type of displaying length means the your query, the your query is the having the less similarity with the this species, or query coverage is, your query coverage is less than this species, right? So so you can easily find out the results of your query sequence. Either it is your query sequence is similar with the other species or not. So for the today, this is the results, and today that is over for this, and we will discuss about the description results, alignment and taxonomy in another video. So that's all over for the day. Thank you so much.